It is important to examine the content of aligned RNA-seq data files before continuing with further analysis, in part because the results of that investigation may, in fact, point you toward how you should best analyze your data. GenePattern provides modules to calculate such quality control (QC) metrics, such as depth of coverage, continuity of coverage, duplication rate, expression rates, strand specificity, and GC content, among others. Having these sorts of metrics can help to prevent or better understand common RNA-seq errors stemming from such sources as read length, quality of data, sample prep, or number of reads in the data. The modules currently provided by GenePattern for this RNA-seq QC workflow are Picard Add-or-Replace Read Groups, Sort SAM, Picard Create Sequence Dictionary, Picard Reorder SAM, Picard Mark Duplicates, SAM Tools Fasta Index, and RNA-seq Metrics. This tutorial will walk through the suggested RNA-seq QC workflow in GenePattern as outlined in this decision diagram. The first input is an aligned, coordinate-sorted BAM file with read group information, such as platform or sample, in the header. If your aligned BAM file does not contain read group information, you should run your data through Add-or-Replace Read Groups, as discussed next. More information about the SAM-BAM format can be found at the SAM Tools website. Note that if your data was aligned with Top Hat, you will likely want to run your BAM file through the Picard tool Merge BAM Alignment, soon to be available in GenePattern, to handle the fact that Top Hat removes unaligned reads. This can throw off the total number of reads and any other metrics using that value in the RNA-seq Metrics module. The first step in this suggested RNA-seq QC workflow is Picard Add-or-Replace Read Groups. Input for that module is a BAM file which has been aligned. The module will either add new read groups if none previously existed, or replace read groups as defined in the parameters. All reads in the file will be assigned to the specified read group. Read group information is required by Picard Mark Duplicates and the RNA-seq Metrics module. Specifically, the RNA-seq Metrics module requires a read group ID in the BAM header. Once the module has finished running, the resulting BAM file can be sent to Sort SAM. To be sure, it is coordinate sorted. Note that the module will also index the file and can be used to convert between SAM and BAM formats, though an index will only be created for BAM files if the sort order is set to coordinate. The next step in our workflow is to index our reference Fasta file. To do this, we'll use the SAM Tools Fasta Index module. SAM Tools Fasta Index takes a reference fasta.fa file and creates a .fai index file for it, which will be used by both Picard Reorder SAM and RNA-seq Metrics to quickly locate and retrieve information from the reference sequence. The module outputs both the originally input Fasta file and its newly created Fasta index in .fai format. The .fa Fasta file can now be sent to Picard Create Sequence Dictionary. This module takes the reference Fasta and creates a SAM file containing a sequence dictionary. A sequence dictionary contains the sequence name, length, genome assembly identifier, and other information about sequences. The sequence dictionary file is required for both Picard Reorder SAM and RNA-seq Metrics. Once the module has finished running, we can send the sequence dictionary output directly to Picard Reorder SAM. Now that the .fai and .dict files have been created for the reference Fasta file, Picard Reorder SAM can be run to order the reads in the BAM file according to the contigs of the reference Fasta file. The module takes as input a BAM-BAI pair, as output from Sort SAM earlier in this workflow, a .dict sequence dictionary from Picard Create Sequence Dictionary, and the .fa Fasta and .fai Fasta index file from SAM Tools Fasta Index. It then reorders the BAM file in accordance with the contigs in the reference Fasta file provided. The order is determined by the exact name matching of contigs. Reads that map to contigs absent from the reference file are dropped. The next step in our workflow is to mark duplicate reads in the BAM file to allow duplicate read metrics to be calculated in the RNA-seq Metrics module. The output BAM file from our recent run of Picard reorder SAM can now be used as input to Picard mark duplicates. As we intend to view the duplication metrics in our final result files, we must leave the remove duplicates parameter at its default setting of no. The duplication metrics will only be calculated if the duplicates are marked, but not removed. Once the module has completed its run, the resulting BAM file can be sent to SortSam to create an updated index. It is important to make sure indexes are always updated each time a change is made to a BAM or SAM file, and that the BAM or SAM file always have an older timestamp than its index. When running SortSam, be sure to select a BAM output and to leave the sort order at coordinate so that an indexed BAM file will be produced. Once SortSam has completed, the BAM-BAI file pair can be sent to RNA-seq metrics. Now that we have completed all of the prerequisite steps, we can run the final module in our workflow, RNA-seq metrics. RNA-seq metrics is the last step in the GenePattern RNA-seq QC workflow. The module calculates standard RNA-seq related metrics, such as depth of coverage, ribosomal RNA contamination, continuity of coverage, and GC bias. It takes the following as input: a coordinated sorted BAM file containing a read group ID which has been reordered by config and optionally has duplicate reads marked, and its associated index. Here, the output from SortSam in the previous step has been specified as input. A GTF annotation file is also required. An HG18 and HG19 GTF annotation from GeneCode are provided in a drop-down list. Other genome annotations must be provided by the user. For our data in this tutorial, we'll choose HG18. Also required is a reference Fasta.fa file as output by SamTools Fasta index. The reference Fasta file index.fai also output by SamTools Fasta index, and the reference Fasta file dictionary.dict as output by Picard create sequence dictionary. Note that in most cases, these last three files will all need to be specified separately, as we did here, because they will likely not be in the same directory. Other parameters to note are num genes, which will determine the number of top expressed genes for which to calculate metrics. By default, we set this to 1000, however this requires at least 3 GB of memory, so you may need to reduce this number depending on the configuration of your machine. Transcript level metrics determines whether to calculate metrics at the transcript level. This is memory and compute intensive as it produces more detailed results, and thus will also produce more output files. By default, we set this to no, and will leave it set to no for this tutorial. GC content file. Providing this file will produce an additional section of results pertaining to the GC content of each transcript. For this tutorial, we'll not be providing this file. Once all of the parameters have been set to our liking, we can run the module. Note that it may take minutes to hours depending on the size of your input data, the number of input files, the number and complexity of your desired output files, memory available, etc. So you may find it helpful to request an email when the job completes. When the job does complete, it will have produced a single .zip archive containing all of the metrics calculated. To view the metrics, download the zip archive and unzip it. In the top-level directory, you'll find an index.html file. Opening this in a browser of your choosing will display a web page containing an HTML report of metrics, stating the total number of reads, depth of coverage at the 3' and 5' end, and more. The report also links to a GCT file containing the calculated RPKM values for each transcript in each sample, and coverage plots for the high, middle, and bottom expressed transcripts. Complete details on all of the metrics calculated are available in the module documentation, which can be accessed by clicking the Help link in the upper right-hand corner of the module page. For more information about GenePattern, please visit genepattern.org.